All right, welcome back. We are getting ready to do the last stage in cellular respiration. That's stage 4 of the electron transport chain. And this is probably one of my favorite parts of all of cellular respiration. Just the beauty of it is pretty remarkable. And what's also cool about this is not only is this happening in bacteria, but it's happening in all of your cells, except for your red blood cells, all the time, every day. And if it doesn't, you're in big trouble. Okay, so we went through glycolysis, acetyl-CoA step, and Krebs. In the process of all of those reactions, those are all catabolic reactions. And by the end of those, we basically completely broke down glucose to carbon dioxide and, well, we haven't made the water yet. We made a little bit of the water, but all of the CO2s were accounted for. For 1 molecule of glucose, there were 6 carbons. 2 of those carbons came out in carbon dioxide in the acetyl-CoA step, and 4 of those came out in Krebs cycle. Each spin of the Krebs produces 2 carbon dioxides. There's 2 spins of Krebs per glucose. So all of those carbons that came in, all those 6 carbons that came in glucose are now exhaled as carbon dioxide as a waste product, which is really cool because when I first started learning this and kind of beginning to wrap my head around it, I'm like, okay, so the food that I eat, carbohydrates, proteins, fats, which are mainly carbon and hydrogen and some oxygen, when I increase my metabolic activity, I'm breathing those carbons out as carbon dioxide. And what can you do to make yourself breathe faster? Well, you can just breathe faster, but then you'll hyperventilate. But that's not what I'm talking about. Exercise, right? So the more you burn, right? You're exercising, your body is burning the fuel. That fuel, the carbon, gets converted into carbon dioxide that you exhale. And that's one of the reasons why you lose weight is you are burning through your fat stores and exhaling all of the carbon that's stored in those lipids as carbon dioxide. Cool. So the more you exercise, the more you do cellular respiration at higher rates, the more you breathe off basically all of your carbon that's stored as sugar and fats. Imagine that. So that's what we've done up to this point is completely oxidized carbon completely oxidized glucose into carbon dioxide. But in the process, we have collected electrons. Remember, we collected electrons in glycolysis, the acetyl-CoA step, and we've collected a lot of electrons in Krebs because we had those redox reactions all over the place. So we have quite a few NADHs and a couple FADH2s. Now what are we going to do with those? That's where the electron transport chain comes in. This is where oxygen is going to play its role, and this is where most of the ATP is going to be formed. And just to note, the ATP that's formed in glycolysis and Krebs cycle is formed via substrate level phosphorylation, just to bring that up. Okay, so here's more of a schematic view of the electron transport chain, and then I'm going to show you a little bit more realistic drawing here. But here are those electron carriers that we produced in steps 1, 2, and 3, NADH and FADH2. And then what they do is they systematically release those electrons, reducing these electron carriers, all of these little guys here, until finally we are going to reduce oxygen as the final electron acceptor. So in each one of these, I'll kind of do a highlighter. There we go. So in each one of these, reduction, oxidation, reduction, oxidation, reduction, redox reactions, the energy that was stored and kind of harnessed by those in those FADH2 and NADH molecules, those electrons are going to do work for us. They're actually going to do some active transport. And we are actively transporting these hydrogen ions against their concentration gradient, which is going to help drive the production of ATP. Okay, you ready? Let's do this. So here's a more realistic view, not just those little kind of circles. So this is going to be a series of proteins and enzymes embedded in a phospholipid bilayer. If you are a bacterium, this is going to be in your plasma membrane. If you are a eukaryotic cell, all of these proteins are going to be embedded in your inner mitochondrial membrane, which kind of goes back to our understanding of the endosymbiotic theory because, oops, yellow is not a good color for that. If you are a bacterium, you are going to have all of these proteins embedded in your plasma membrane because that's the only set of membranes you have. Right? So let's say you are a free-living electron transport chain doing prokaryote, right? Then you get engulfed by a larger cell. And so now you are inside this little vesicle. So all of your electron transport chain proteins, so this guy kind of gets engulfed, you are now inside this other vesicle. And over millennia, millions of millennias, to increase your efficiency, the outer membrane kind of stays about the same, but now the inner membrane gets really folded. So that's the inner membrane now. The more folds you have on your inner membrane, the more sets of these electron transport chain proteins that you need, so you become really efficient. So this idea that a free-living bacterium with its electron transport chain proteins embedded on the plasma membrane evolved into a mitochondria with the same electron transport chain proteins embedded in the inner membrane really supports that idea of endosymbiotic theory. I just love that. I love that connection. It's so amazing. All right, so this is what we're talking about. If you are a bacteria, all of this is taking place in your plasma membrane. If you are a mitochondria, it is taking place in your inner membrane, on your inner membrane. Either way, it's all the same. So the NADH, the FADH2 that we've just created in steps 1 through 3 in cellular respiration get shuttled into either the mitochondria, if you're a eukaryotic cell, or you're just hanging out in the cytoplasm because that's where you are. You will then come to this first protein complex. And that NADH plus complex number 1 goes to NAD plus, and let's see. Complex number 1. I'm kind of giving it away here because I'm going to put the electrons with the complex number 1. So we have the electrons hanging out with the NADH, and they go through this reaction, and they are no longer there. So now who's getting oxidized and who's getting reduced? So remember, losing electrons is oxidation. So NADH is oxidized now. They were just temporarily holding on to those electrons. Remember, all the NAD pluses were reduced into NADH. Now all the NADHs are going to be oxidized, and the intermediate, these complexes are going to be reduced because they're going to be gaining those electrons, even temporarily. It's that kind of oxidation reduction like we saw in the last schematic. All right, so that's our step 1. NADH is being oxidized, it's losing its electrons, giving those to complex number 1, which is then going to allow the movement of those electrons to pump protons. So these, a black highlight does not do a good job. So we're pumping protons against their concentration gradient. That's active transport, but we're not burning ATP to do that. We're using the power of those electrons. We have another redox reaction here with FADH2 going to ubiquinone. FADH2 is going to be oxidized, ubiquinone will be reduced, and we're going to move those electrons to complex number 2, which we're going to, again, oxidize and reduce, pumping more protons against the hydrogen ion concentration gradient. We move those electrons down another carrier, the cytochrome C, to the final carrier called cytochrome C oxidase. So again, we're going to be pumping more protons, these hydrogen ions, against their concentration gradient. And our last electron acceptor, because these electrons have to go somewhere. So they go and bind with oxygen. That's what we take in, right? So that's what we're breathing in. So if we do our cytochrome C, to a pen mode. Cytochrome, cytochrome C plus electron, nope, it's getting on towards the end of the day, plus our oxygen, 1/2 of our oxygen goes to O2, and water. So who's getting oxidized and who's getting reduced? Well, who's going to be gaining the electrons? The electrons are going to be, no, let me back up, sorry. Let me just write that all over again. We have cytochrome C has the electrons, plus half of the oxygen goes to water. Because the cytochrome C is it's not changing any of its electrical or chemical makeup because it's just a stable protein. It's not like a succinyl-CoA or a malate or anything like that. So the cytochrome C is actually what has the electrons to begin with, and who gained the hydrogens. That was our clue that we're looking at the whole time. So that tells us that cytochrome C is oxidized and the oxygen is reduced to produce water as a byproduct. So when we breathe oxygen in, the sole purpose of you breathing oxygen is so that it can be this final electron acceptor with a couple hydrogens and make water. That's the bulk of the water that's produced in that original equation that we started at the beginning. So, okay, we just took care of all of those electrons that we collected along steps 1 through 3, NADH and FADH2, dropped them along this electron transport chain. As they were pumping along, they pumped the hydrogen ions against their concentration gradient. Now what? We still haven't produced any ATPs in the electron transport chain. Well, what we have set up is a huge hydrogen ion gradient on the one side of the membrane. Now, remember, membranes are impermeable to hydrogen ions because of that hydrophobic middle part. So they just can't sneak back through. So what our cells have, and bacterial cells and fungal cells and all cells that do this aerobic respiration, is this wonderfully awesome enzyme called ATP synthase. It is an ion channel that allows the hydrogen to go back down towards its concentration gradient, right? Facilitated diffusion, that's all we're talking about, as the hydrogen ions travel back down, trying to create an equilibrium. But the nature of this enzyme is that as those hydrogens are traveling down their concentration gradient, it mechanically spins this part of the protein. And as it's mechanically spinning, that mechanical energy hooks that third phosphate group onto the ADP, which is a very endergonic reaction, but facilitated diffusion is an exergonic reaction going from high to low. So that exergonic reaction drives the endergonic reaction coupling, for those of you who might remember that from Bi 211, to produce your ATPs. This is what we call oxidative phosphorylation, this redox, producing the gradient, chemiosmosis driving the ATP synthase, phosphorylating the ADP to give you ATP. It's so beautiful. I love it. I'll be posting some animations also with this chapter 5 information. So if you wanted to see it I would show animations in class, but I can't do that during the way that this is set up. So go and click on some of those videos and it might help kind of seeing things in action. Here's a video. No, well, it was supposed to play. That little GIF is supposed to play, but it doesn't look like it's going to. It might on your version. Oh, there it goes. Yay. So here is a space filling model of the ATP synthase right here. And then here is the little animation showing the little yellow circles coming in from the top. Those are the hydrogen ions, right? So high hydrogen ions down here, low hydrogen ions here. So we have a concentration gradient. The hydrogen ions want to go back down their concentration gradient. As they travel through this specialized channel, it physically rotates that bottom piece, the lollipop head part. And these little kind of card looking things that are flying in here, that's the ADP in inorganic phosphate. And the red thing leaving, that is the ATP. So ADP plus PI go in and ATP comes out. And there's 3 slots. So it does this, each kind of partial spin allows the next stage to go in. It's so cool. So I encourage you to look up videos and animations on all of cellular respiration or the electron transport chain if that's the part that's giving you trouble, or the ATP synthase molecule itself. And that wraps, oh, let's see. No, I have a summary. There we go. If we're going to go back and count all of our ATPs, like the accounting of how much ATPs do we get per molecule of glucose, it's about between 36 and 38, depending on which kind of cell you are and which resource you have. Some actually say 34, but we're going to say 38 for our bacteria. We get 2 net ATPs from glycolysis, 2 ATPs from Krebs, remember the GTPs being converted to ATPs, and we get 34 ATPs from oxidative phosphorylation in the electron transport chain. So all those electrons that we harness in NADH and FADH2, those are responsible for producing the bulk of the ATPs. So if you're thinking, why do I have to learn all of those redox reactions? It's because those are what keep you powered. Those NADHs are what produce 30 out of 38 of your ATPs per molecule of glucose. Super amazing. All right, that wraps up our cellular respiration. We'll be doing some alternatives to glycolysis, fermentation, and photosynthesis to wrap up chapter 5. I will see you next time. Bye.